That's having an issue with TV.
Yeah.
Your audio is fine.
Oh, let me turn it
I got it.
down, yeah.
There we go.
Okay.
Can they see on Zoom?
Is my audio going through the owl or through this computer?
Don't know.
You're muted on it.
Oh, okay, so can
Turn it.
Alex hear me?
Alex, can you hear Joseph okay?
The presentation mode isn't shared.
Or is the Zoom?
Yeah, I could hear you guys and I could see the presentation.
Okay,
Okay, cool, thank you. All right, that was quite the hiccup. Thank you guys for waiting. That's not supposed to go. The research that I wanted to talk about today is the design and synthesis of novel agonists for the treatment of gut disorders. And yeah, let's get to it. So let's try to make this somewhere else. Be down here. Sorry. Okay, so the introduction that I wanted to introduce to you all is the disease that we're going to be talking about today, inflammatory bowel disease, or IBD. This is really bothering me. Is there a way to do it if I do it?
And you're still not sharing on Zoom, I think.
Alex said he could see it, I think.
It shows the title slide right now.
Yeah, I think
Okay.
it's the other view.
Can,
You pause your
Wish you
share.
can share.
okay.
Maybe stop and start again.
Stop it.
Yeah, I'm sorry.
I'm so bad at computers.
Try sharing the whole desktop instead of the like presentation.
Okay.
When you share.
Now you're, where are you now?
Oh, well, I have to share down here. Desktop.
Yeah.
Okay.
Can everyone see it now?
We can see your screen now.
you can?
Yes.
Yes.
Okay, thank you.
All right. Okay, so as I was saying, inflammatory bowel disease. Some really interesting notes is that the average age of diagnosis for IBD is actually very young. It's 21 and it's a chronic disease, so you have it for the rest of your life. And on average, patients have 2 surgeries, so it's actually very life altering. It affects people's lives a lot. And currently there's 1.6 million Americans today that have it, 1 in 200 people. So there's a lot of people that have Crohn's disease or colitis. It's actually a very complex disease. You can see there's more than 100 genes contributing to IBD risk. And so currently the main treatment for IBD is found in biologics. You can see it takes up about half of the costs for IBD. And of those biologics, most of the biologics are TNF alpha blockers. So TNF alpha is an inflammatory related protein. And this statistic should show you that the main issue why we want to target IBD, up to 40% of Crohn's disease patients treated with TNF alpha blockers become refractory after just 1 year. And this is, again, a chronic disease. So we have patients that have this for their entire lives. And that's quite a large percent of patients not able to be treated with the main treatments that we have today for IBD. So to circumvent this, the collaborators that our lab is collaborating with are heavily involved in trying to find targets for IBD. So they used an AI-assisted target identification system. And so I'm going to try to walk through that with you guys. First, they started with a large data set of Crohn's disease patients and their RNA data. And they walked through the different genes that are in healthy patients and the different genes that are in Crohn's disease patients and how those genes are related. And using a Boolean algorithm that relates those two genes, they created clusters of genes that are similar. So here are some of the relationships. For example, if a gene A is high for this green arrow, and gene B is high as well, and so on. And so those clusters are then mapped out in this large data set. And then a pathway from healthy to Crohn's disease is mapped out with these different clusters. So for example, in cluster 1 of similar genes, you may have a high expression level in healthy patients, but a very low expression level in someone who has severe Crohn's disease, and maybe a medium expression level in someone who has symptoms, starting to have symptoms of Crohn's disease. So it's a kind of a continuum pathway. So here are some of the clusters shown, and there's quite a lot going on, but I want to show put your focus to cluster 2 and cluster 6, and these two clusters contain 2 genes that are going to be the target for my talk today. PPARG and PPARa are the genes that encode for PPAR-gamma and PPAR-alpha, and they're found in cluster 2 and 6. And these are both nuclear receptor genes, as well as or proteins, as well as proteins that are heavily involved in the metabolism of our bodies. So just to give an overview of what the collaboration looks like and what my part is in this collaboration, and the part that the Ghost Lab is doing is, as I've shown, they've used Boolean networks to analyze gene clusters and have a target identification. And so PPARa and PPARg are those targets for inflammatory bowel disease. And then I'll go ahead and show some of the molecular docking that I've done in silico, as well as using crystal structures that have already been published. I'll show some of the chemistry that I've been doing to try to make novel molecules. After that, I plan on doing in vitro protein assays to characterize the binding of these novel molecules. And they'll go ahead and test these molecules in vivo, as well as they have a system for using patient-derived disease organoid models to validate the target identification, and then goes on to clinical investigation. So that's the idea mainly. So I wanted to give some background for what is PPAR, what does it stand for? It's a peroxisome proliferated activated receptor. And shown in this figure, you can see that there's lots of different ligands that bind to the PPARs. There are 3 isoforms. So as I showed before, there's the alpha and the gamma, but there's also the delta, which I won't be talking about today. They are nuclear receptors, and so they find themselves in the nucleus of the cell. They're also transcription factors, so they bind with retinoid X receptor, or RXR. And so upon binding to the PPAR response element, or PPARe, on the DNA, these PPAR alpha or PPAR gamma RXR dimers, they activate target genes and downstream signaling. And so that controls a whole host of things. So for example, the alpha dimer will control peroxisome proliferation, it will control fatty acid metabolism and lipid homeostasis. And for the PPAR gammas, that with the RXR dimer, it will affect adipocyte differentiation and macrophage function. And so I'll get to it a little bit later in my talk, but note that the ligands that bind to PPAR gamma are traditionally called glidosomes, and the ones that bind to alpha are traditionally called fibrates. And another figure just to show that they're involved in a whole host of different metabolic functions, including, you can see, inflammation in both alpha and gamma. So this next slide is to show that the balanced dual agonism of PPAR alpha gamma is critical for the treatment of IBD. And so we've shown that we've been able to publish using a known molecule, PAR 5359 shown right here. It's shown to have a much more bacterial clearance and better results than the PPAR alpha agonist or PPAR gamma agonist by itself. So drugs that bind to both isoforms as well as have the same or similar activity are the drugs that our collaborators have shown to have the best results for this disease. And you can see here that the known molecule has roughly similar activity with the EC50. So that can be explained by a number of things, but what we predict is that the alpha is involved in inflammation, and if you target it, it's actually going to induce a bit of inflammation, but it's going to have the very important effect of clearing microbes. So having all of that bad bacteria cleared out is very good. And so coupled with the PPAR gamma, that's the healing or the resolution of the inflammation. And so having them balanced together is the key. So the current scope of PPAR therapeutics is that there's actually not that many drugs that are approved in general for PPARs, and that all of them are approved for type 2 diabetes. So targeting IBD with the protein PPAR is actually a very, very new idea and new concept. These PPARs have been discovered since the 90s, but they've generally only been targeted for type 2 diabetes or similar conditions such as dyslipidemia. So you can see here on the right some of the molecules that have targeted these PPARs. And so the top right are the PPAR gamma specific molecules. And I'll show you guys in the next slide that they all have a very similar head group, which makes them the glitazones. And then the next one is an alpha specific molecule, and it's got a specific head group as well. And it's a prodrug, so this will be carboxylic acid. And then these glitazars are the alpha and gamma dual agonists, and they also have a very specific head group, as you can see. Not all of them that bind specifically to gamma or alpha have these head groups, but most do. And so here are the head groups that I was showing and how they affect PPAR selectivity. And you can see that PAR 5359, which is the molecule that we published on that showed really good results for bacterial clearance, has that alpha gamma dual glitazar head. So the specifically the S 2-ethoxypropionic acid. Yeah, so for the next step, what I did is I wanted to see how is it balanced? Is there any way we can try to figure out how this molecule is balanced, and how might it bind? And so I used docking studies to help me out and show me how it might bind. It's not a crystal structure, so it hasn't been crystallized, so I don't know exactly how it binds, but these docking studies give me a little bit of a glimpse of how it might bind in the pocket of these PPARs. So I went into MOE, a docking simulation software, and I took the X-ray crystal structure of this molecule, rosiglitazone. It's the most well-known PPAR gamma selective molecule. And I docked both. And you can see here that the two histidines in the PPAR gamma pocket hold on to the gamma selective molecule, this TZD group right here, to the two oxygens, kind of like a bike handle, and it's very tight binding, as well as it fits very well into the rest of the pocket and even has an arene interaction. And so the PAR 5359 does a similar thing. I went ahead and docked the alpha selective molecule into the gamma pocket, and fortunately it gave me the results I expected, which is that it's not very selective. it doesn't fit well into the pocket, and it doesn't have those hydrogen bonding interactions you saw previously, which are really, really important and a major driving force for good binding. As well, then the next thing is I docked the PAR 5359 into the alpha pocket shown on the right, and the green alpha selective molecule also showed good binding. And the gamma molecule that's supposed to be selective for PPAR gamma wasn't able to make hydrogen bond interactions as well. I also looked at X-ray crystal structures of these molecules to see how a dual agonist might actually bind. So these are not docking simulations, but this is the actual X-ray crystal structure of both PPAR gamma shown on the left and PPAR alpha. And you can see that tesaglutazar, it has actually very good balanced activity, but its activity is not as low as one would like, and it's actually was on the list of the previous slide of molecules that are too toxic and didn't make it through clinical trials. But you can see here that it's very important for it to fit into the pocket well with the linker and the hydrophobic region, as well as the hydrogen bonding interactions. They're very similar in both pockets. So for my predictions in my research, I have two hypotheses. One is that the ether oxygen right here in the molecule and the acidic oxygens play a key role in having this ethoxypropionic acid moiety bind to both proteins. So I show that here with this is the same docking simulation as I showed previously, but in a 2D form. And in the alpha, so this is the alpha pocket, the histidine will hydrogen bond with that ether oxygen. And in the gamma, it binds with the, it interacts with the acidic oxygens here, not shown, but that is one of my hypotheses, is that both these oxygens there give it flexibility to be a dual agonist. And then of course, a well-spaced linker is very crucial for lining up and fitting into that pocket, as I showed with the alpha not binding into the gamma and the gamma not interacting very well with the alpha. So here are some of the designed molecules that I have. I can't move it. Okay, that's fine. You don't need to see the last one. Yeah. So yeah, some of the molecules I have designed are changing the head group to see if that does much, or changing the oxygen in the ether group to a nitrogen to see if that changes binding. This last one is basically the alpha selective head group that I showed previously, fitted onto the PAR 5359 scaffold. And this molecule here is taking out the tetrahydropyridine and putting back in a benzene ring to see if that affects any arine interactions. How am I doing on time? Okay.
Wrap it up if you can.
Sure. Yeah, the next couple slides are just the synthesis of these molecules, and I understand like a lot of people in interfaces are not specifically in the chemistry field.
You made it.
Yeah.
It's fine. You don't move on.
Yeah, I made all the molecules. Sounds good. Yep. Okay. And then the next step is in the Ghost lab, they tested some of the molecules that I made, and they used a reporter assay that created a luminescence, and the luciferase detection reagent is directly proportional to its activity. And there were some interesting data shown just for this initial molecule that I gave them. I also gave them JS2, which has a nitrile, not shown, but that was not as active. And it has a 1-to-3 ratio for its activity, so it's not balanced. And for the next steps, I hope to find out if the other molecules that I've synthesized are more balanced or not. And And yes, some of the next steps are to do ITC binding. So that stands for isothermal titration calorimetry. And here you use an instrument that injects a solution of the ligand into buffer and protein here in the sample cell. And it gives a curve of the heat released. So better binding, more heat. And you can relate that with some formulas to give you the affinity, give it the stoichiometry and essentially the binding, how well it binds, or the KD. So this on the right is an example of ITC data for PPAR, and these people publish using resveratrol, which is actually a natural product found in wine, I believe, and other natural sources. And yeah, they show a certain curve, and then you can use the Michaelis-Menten equation to get the KD. So that is also included for my next steps.
You don't need to tell them the equation.
Okay. And yeah, that is what I have. So for some of the next steps that I want to do for my research is doing X-ray crystallography, because I want to validate my docking studies with what it would actually look like if my molecules were bound to the protein. And I really want to compare the binding affinity to the activity to see how are they related, because that's an interesting question that we have. So yeah, just want to wrap that up. And I really want to acknowledge the Interfaces Training Grant. That's really been helpful, and learning a lot about other people's research. I really want to acknowledge the Ghosh Lab that I've been collaborating with since the start of my PhD, and especially Professor Ghosh and Dr. Gaj, who have been really helpful in testing some of the molecules that I've synthesized.
Have you participated in those in vitro experiments yourself?
I am currently right now working on the ITC experiments. Yeah.
I also forgot to mention, you had to express both the PPAR alpha and the end, so
Yes.
what you have done, it's done.
Mhm.
That's an important thing to give yourself credit for things that you've actually accomplished out of this. Try to coach on everyone involved.
I mean, being able to say you did the theoretical work and the experimental work will pay off one day.
Yes.
I think, so I'm a nutritionist, and I think treating IBD is one of my favorite conditions. Just because there's so many different angles in which you can approach the condition, depending on state, severity. And I started to giggle a little bit when you showed it on resveratrol, because a lot of people think the benefit of wine is that it has resveratrol, which is true, but you get the same benefit from grapes because it's just loaded in rich grapes. So you don't have to drink wine to be healthy. You can eat grapes. Actually, there's more resveratrol in the grapes because of processing. But the reason I bring that up is because when I think about inflammatory bowel disease, you have a lot of material to work with. And when you activate PPAR, for example, you can do it through stress reduction. It's a big one for some people. Other ones, it's food. Obviously, we've talked about inflammatory foods. But I'm curious to know, with respect to like drugs' ability to impact the microbiome. We know that a lot of IBD patients have alterations in the gut microbiome. And I'm curious to know, like, would you consider pursuing maybe just like a little organoid model of like how one of the drugs that you're synthesizing may have like an impact on the composition of the gut microbiome as well? Just because like antibiotics are known to impact the gut microbiome, obviously, but we know that other drugs have subtle modifications too, which may attenuate some of that inflammation seen. So if it's activating the PPAR, for example, via one mechanism, is the drug also impacting the drugs via another mechanism, which is contributing to the beneficial side effects of IBD?
Mhm. So if I understand your question, like, I'm not sure if I do, but like,
Is
you're
the-
asking, yeah,
Is the drug having an impact on the microbiome?
yeah.
Microbiome, essentially. And
And
is
you're-
it beneficial?
like how to test that with,
Yeah, or just like what happens like pre-post administration?
yeah, I think that's certainly something that maybe I don't have like the capacity to pursue. That's something that like I could bring to my collaborator and like Dr. Gaj, and they have plans on doing like organoid models based off of patients that have Crohn's disease. So yeah, I don't have the expertise to answer whether that, like, I don't even know how I would go about testing that, but that's like a really interesting question.
They can test the gut on the mice. You know, people or the control,
Yeah.
the treated mice, to see how it affects the gut, the microbiome of the gut, which is highly dependent on colony, I mean, and which building they're in. It's like there's so many factors that affect the microbiome. That it's actually.
And
So you know Larry Smart, who was the director of Qualcomm Institute, so he retired last year. He has Crohn's disease, and he actually would sequence his entire gut microbiome like every week. And actually would correlate the changes in the microbiome composition and diversity with the flare-ups in his Crohn's. And so it's actually a big data set somewhere around here.
Yeah, so I have two questions for you. So one, why use a balanced dual agonist rather than just take an alpha and a gamma, each selective, and add two drugs?
Yeah.
That's one question. Why not?
I think my understanding is that, or at least what Dr. Gaj has told me is that if you do that, and I guess you try to do it simultaneously, they don't have a simultaneous effect or activity of the two proteins, and so then one might win out on the other and give a lot more effects of like, if the PPAR alpha gets activated, then maybe there's too much inflammation. If the gamma gets activated, it's not going to give the bacterial clearance that's desired. That's my understanding of like that they're not going to be activated at the same time, I guess.
Yeah, I mean, I think that's kind of what I would say is that it's hard enough to work with PK and PD of one compound.
Yeah,
Then you gotta do two acting simultaneously. That is probably just doubles or maybe even exponentially affects the drug, measure we have to use. Another question is, what is balance? So, you have a 3-to-1 binding or 3-to-1 EC50 right now, right? And so that's, I mean, then the order of magnitude is not good enough for what do you think is balanced? Did the Ghosh Lab mention to you, well, what is our target in terms of balance?
yeah, I'm meeting with Professor Ghosh this week to discuss that exact question, actually. I think from my understanding, it would be somewhere around like between 1 to 2, or at least
Less than 2-fold.
yeah.
Okay, so we're at 3-fold, so double what you did. Gotcha. Okay.
Thanks, Joseph. That's very nice. Got to hand over to Danny.
Thank you, Joseph.
I didn't think that was.
Started, I'll be presenting an epidemiology project that I've been working on for this past year with our athletics department. So a bit of introduction, there are approximately 8.6 million sports injuries in the US per year, with about 3 million of those leading to an ER visit. So though sports injuries typically have a pretty low mortality rate, there's a lot of secondary issues that arise from these injuries, secondary injury and disease, recurrence of these injuries, chronic pain, and then the cost associated with these issues. There's also a lot of longer term problems that can arise. A study of UK soccer players found that the risk of acute injury in professional players is up to 3 times higher than those working in construction and service jobs. And then similarly, there's also a high prevalence of arthritis in professional soccer players' knees, and a lot of other diseases based on the sport that's being played. So in the existing literature, there aren't very many papers that have a broad look across multiple sports and multiple years. But there is a lot of activity directed at injury prevention programs, return to sport programs, and training programs. But all of these programs require a good long look at what the injury rates actually look like across time to see where we need to be focusing our efforts. So that's why we decided to do this study. So we took 10 years of data that UCSD has been taking on athletic injuries and divided it up by body area, across time, by gender and sport, to examine where we should be focusing our efforts. So I basically said this, we had over 5,000 participants over the 10 years with over 6,000 injuries logged across the 23 sports that UCSD has. All of these injuries are logged by the athletic trainers, which attend all practices, games, and workout sessions. And then the more severe injuries see our 2 sports physicians. So all the data was taken from the 2 databases that was taken across these 10 years. So to start, just looking at the normalized injury rates for all the sports. What we're looking on the y-axis is the average yearly injuries for the sports divided by the number of athletes for that year. And you see there's a pretty big range with some sports being under 0.5 injuries per athlete per year, with some going as high as 1.6. And then depending on the year, as high as I think 2.37 was one of the highest. And at least for me, this range was pretty unexpected, seeing that some sports have over 1.5 injuries per athlete per year. So on average, they're getting injured more than once a year. Then looking at these same rates for each sport by year, you see that it's pretty level. There are some spikes, which we're working with the athletics department to identify maybe what happened, maybe why some years had almost 0. This is also in the way I can't remember. This is, yeah, the women's sports. And then we have the same thing for the men's sports. We then decided.
Is there a sort of a natural experiment during COVID?
So yeah, I was going to talk about this right now. So we divided up by body area with miscellaneous, as you can see in like all the way from 2019 or to the 2020 spring season up to 2022. That's pretty much all of what that miscellaneous is. So the rest of the graphs that I'll be showing won't be including these last 2 years, but I am talking to the athletics department to see what data we have exactly on COVID and how practices were impacted, to see what kind of natural experiments arise from having that data. So here we can see it by sport and by body region with the total number of injuries over those 10 years in the middle. You can see that lower extremity on average is the highest for each of the sports, which makes sense for things like cross country and track. But we really see that the men's and women's teams have similar injury breakdowns in pretty much all sports. We see slight differences in some, like baseball, softball. But overall, they're pretty much the exact same breakdowns, which we can see even deeper here. So here I divided it up by those body regions to see what % of the total injuries are for each of those body regions by sport and gender. And then we're also looking at, again, the outliers here to see what maybe happened those years. And then another thing that we're really focusing on right now is comparing the injury rates to the training hours for the athletes, because in every sport that we've looked at so far, whenever there's a spike in training hours, we see a spike in injury rates, which makes sense intuitively, but we also have to examine what we can do about that to reduce those injury rates. So I have a few examples here, like men's and women's volleyball, which have different seasons, but still see that spike with the increase in training hours. And then again, over here in the season for men's volleyball. And then the same thing in women's and men's soccer. And in basketball. So regardless of whether they have corresponding seasons or not, we see those spikes. And also working with the athletics department on this definition of severity that we've created, so Allison Pastor is our director of rehab, I think is her title. She's gonna be going through a year and trying to categorize all of our injuries with this new severity scale that we've developed to see how to see the different severities of injuries and again, where we need to focus more on those higher severity injuries. So I'm still working on statistical analysis. I have some of the ANOVAs done. I did everything again in R. We're still comparing those training hours to injury rates, severity, and also looking at the top 5 injuries for each sport to then do a deeper dive on those specific injuries. As opposed to just a general sport specific.
The one thing, is ANOVA the right analysis to apply to these numbers?
That's what I've been looking at. Sam thought I should start there, just to get a better idea.
I mean, at least to sort of learn the techniques of
Yeah.
calculating, but I think when you talk about incidence,
Mhm.
so anything that's like a fraction or an incidence or a risk or a probability, which is basically what these are.
Yeah.
As opposed to a sort of a numerical distribution, I mean, is usually it's binomially distributed, and we usually use a logistic regression.
Mhm.
In other words, it's like ANOVA, but you're basically working on the logs.
Yeah.
So the sum of the logs is product, and that's because, you know, the risks sort of, if you have a probability of one thing and a probability of another, they combine as a product. So I guess it's the right analysis for these type of data is probably logistic regression, not
Yeah.
ANOVA, but I mean, the way you use them is very similar, but essentially the main difference is that it uses logs of the numbers.
Mhm.
Anyway, something to read about.
Yeah, I mean, that's really what I had to share for today. I had like a couple slides with the ANOVAs on there, but I kind of mentioned the highlights.
I have a question.
Yeah.
Because this is eventually gonna come up in my research as well, but when presenting data on 2 different sexes, and remembering that gender is a social construct, how do you identify with athletes or patients who don't identify as man or woman? And like, how does that impact statistical analysis?
I think because, at least in this, all the teams are divided by like men's teams and women's teams. It's just the people who are on those teams. I think we probably have to have another conversation how we want to present that data, especially on like when we're talking about specific sports. But it's also all de-identified, so I wouldn't know any of that on any of these participants.
That makes sense. I think when I'm just presenting data, I try to say like sex over gender.
Yeah.
Just because then it just automatically excludes people who don't identify as either one. So just trying to be more mindful of that, and then remembering that male and female are adjectives, not nouns.
Mhm.
So I'm practicing.
Yeah, it's a good point. I mean, it may also be that you can get some clarity by looking at NCAA rules. I don't know if there are any.
I'm sure there are.
If you look at the rules, there are. And they probably differ by sport. And there could actually be very valid reasons for that. So it could be that there may be some way to get some insight by looking at differences in sports based on the rules.
Mhm.
Self-analysis, if you have that information.
I don't think we have that information probably recorded, especially, definitely not for 10 years. But I'll talk to the athletics department, see.
That's a good question.
I mean, we're gonna see more and more of it, I think, as time goes on.
Yeah.
It'll have to be factored into analyses, because again, I think our only, like, our best analysis can only come from like the best intake questionnaires.
Yes,
Because my first rotation was on looking at oxalate data in the microbiome. And the questionnaires in which all of that statistical analysis was done on didn't capture that. So I'm like, what can I do with this? So I think, yeah, these are ongoing conversations that I'm just trying to be mindful of.
so I don't know how much you can do this because you obviously don't have, there's no such thing as a paired comparison between men's and women's sports. But there is, if you were to look at the data that you showed, there's a bigger variation between the sports than there is between the sexes.
Yeah,
And that's probably not surprising. But what I'm wondering is whether there's a statistical analysis that would account for that correlation. And in other words, you can't plot point by point, but you can at least plot, you could plot those same points you plotted, those same means and errors that you plotted against each other, right? And they would correlate. But some of the points would lie above the curve and some the unity line, and some might lie below. And so then you could ask the question whether sort of accounting for the differences across sports. Were there systematic differences, like were there sports where males were more likely to be injured than, significantly more likely to be injured than females, and vice versa?
yeah, that's interesting.
Because for the most part, they're pretty tight.
Yeah, they were.
But perhaps you could use that variation to then, if you could partition out that variation, and it's a little hard because you only have an aggregate, but there might be a way to.
I think the bigger thing we're gonna see is looking at the different training hours across the genders and different types of training that we see. So here we see like the men's team has significantly more time in the gym.
Yes. So when you talk about spike, that kind of implies a sudden change. So the other interesting question is whether there's just a sort of an overall, the same thing, is there just an overall correlation with the injury rate and the number of training hours? Or is it something more than that? Is it like the rate of change of the number of training hours to the injury rate, right? Spike almost suggests that if you change the training too rapidly, then you could get a sudden increase in injury. Whereas if there are other sports where training hours change more slowly, maybe
Well, I think, I don't know if it's one of your examples here. It's not. There's a couple of sports that ramp up
yes.
more like this, and you do see, if you actually look overall, the incidence here is lower than like in soccer.
So again, you might be sort of factor out how much is a difference. It's just sort of proportional to the amount of time spent training.
Yeah,
Versus how much is a difference that's due to particularly an increase.
well, and then you also see that incidence goes down even though the training hours really don't go down.
That's right, yeah, exactly.
Yeah, I think that's a super good question to look more into. Also interesting that for men's and do they play in the same season?
So volleyball has a different season.
Okay, yeah, that makes sense. Yeah, that is interesting.
So how do you think you're going to turn this into this is a sort of a population scale analysis.
Yeah.
What's your idea for making this multi-scale?
I think the logical next step at this level, like we've been talking about, is talking to Rose and seeing what kind of AI models for predicting injury we could maybe develop. And then I've been kind of leading this data analysis, but we have a few other people who are very specific in diving deeper, are very interested in diving deeper on specific sports. Like, I'm interested in looking at water polo and those specific water polo injuries. And then looking at the top injuries for those sports and creating models, probably injury specific models.
Yeah, I mean, I think in a way, if you think about this, it's a sort of the one implicit skill, there's timescales obviously here, which are quite large, but the other scale that you get here is you talk about injuries to the individuals, and once they're injured, they probably, in many cases, they can't do anything.
Mhm.
But then there's also, what the nature of the injury is. So there's a sort of an organ scale, and as well as a population scale, is kind of a limb scale question. So there might be some interesting analyses that you could find there.
Or even preventative like work. I think if I was an athlete, I just got accepted into UCSD, and I was going into baseball, for example, and just knowing that there's a higher risk of upper extremity injuries, like I want to be aware of that, and then want to like monitor the symptoms throughout, just to like how can we predict as soon as possible that things are starting to I'm setting myself up for an injury, essentially.
We're also working to compare the rates that we have with the NCAA. They publish some annual injury rates and seeing if there's areas where we're doing better, doing worse.
I mean, I do think if you find that there is some, that there isn't just a correlation between training hours and injury rate, but there's literally a spike, that there's a difference between increasing the training and decreasing, for example, that you might then be able to design some interesting studies, because you could then ask, is there something before the spike occurs, but after the training has ramped up, is there something you could measure that could be a predictor? Right? So in other words, you could design a study knowing when to look, right?
Yeah.
And what sort of risk factors to be looking for. The other thing I was thinking of is there's this system called that they use over there that uses blood flow restriction. And they mostly use it for recovery. So the sort of the dogma is that since they can't exercise, you can sort of passively exercise your limbs by restricting blood flow and then relieving it, and you get this kind of hyperemia. And so you simulate the kind of ischemia and higher blood flow that occurs when you put exercise demands on muscles. So the philosophy is that they tend to use it a lot for recovery. Some people use it to augment training, because they think that if you do a squat or you do a lift or something, do the same thing while you're undergoing ischemia, that it's an even more intense, intensive exercise. I'm not sure. I mean, it hurts a lot more, but I'm not sure the validity of that. But in the heart, they did this type of thing in the 80s, where they would fire off the coronary vessel briefly, like for 5 minutes, and then release it. And that actually activated signaling cascades that were protective against myocardial infarction. So if in the next few hours, you gave that same heart and then a myocardial infarction, like you tied the vessel off completely, the size of the region of the muscle that died was actually much smaller. They call it ischemic preconditioning. And so it's not terribly useful clinically. It's not like you can go around and tie someone's coronary artery off every, 4 times, twice a day or something, just in case they have an MI, right? You learn a little bit about protective signaling mechanisms, but
exercise has the same impact. Is that what you're saying?
no, I was wondering where the blood flow occlusion does. So if during this time when they're at high risk of injury, they were to have, let's say, morning and night, or morning and just before training, they were to do blood flow restriction, and it might only have to be for 5 or 10 minutes. Then it would be really interesting to see if that group when inevitably some fraction of them are going to be injured. Right? And you could choose group where the likelihood was that was like lower limb or something, this upper limb. You could even do a kind of contralateral control where you only did the ischemia in one leg, or an arm. But you could theoretically then test the idea that it could be protective if the severity of the injuries was less.
I have a question. Maybe go back to your like first or second slide. I was very curious. Oh, no, that one works. I was curious, like about golf, to be honest. That's where, like, are you interested in looking at the statistics and like how do people get injured, considering like it's non-running and non-contact?
I haven't looked much at the specific injuries in golf, especially since like again, over 10 years, they've had 50 injuries, but I'm guessing they just go in and see the trainers for something like shoulder pain or wrist pain, like, yeah, elbow.
And is this only NCAA sports?
Yeah, so we have data on the club sports here as well. But we're focusing on the NCAA sports because we have their actual training data and they have set training hours and we have more information on the athletes in general.
I mean, the involvement of upper and lower extremity definitely follows a pattern that isn't surprising. Higher in baseball, but upper extremity and higher in track and field below that. Yes, thank you for that. Sawires. Okay, well, thanks very much.
Thanks, everybody.
I guess it's our last one for the year.
Yes.
We will